All right, let's get started. Hello, everybody. Welcome to BugFest. You've made it to probably the largest virtual celebration of bugs anywhere ever. At least I've never heard of a bigger bug party than the one that the North Carolina Museum of Natural Sciences has been throwing all week. We've got 6 days of bug science, bug crafts, and activities. We've been doing all kinds of great fun things and learning all kinds of great, amazing things about the world of arthropods all week long. We started first thing Monday morning, and we have more presentations scheduled today and tomorrow. So if you're just now finding us, don't worry. There's still a lot that you can do. Visit bugfest.org. There you can see where to sign up for programs. It's all free. So we hope that you'll join us for more of our programs coming up later today and again tomorrow throughout most of the day. Of course, I'm so glad that you could be with us right now. I'm your host for our programs. My name is Chris Smith. I'm curator for the SECU Daily Planet Theater at the Museum of Natural Sciences in downtown Raleigh. I want to thank our sponsors for BugFest this year, Terminix and BASF. Now, for this program, I hope that none of you are too squeamish because I'm very excited about this program because we're bringing a very interesting story of insects and bugs with a very special guest. So on the show today for our Noontime BugFest program, we have Dr. Ann Perez. Dr. Perez is a forensic entomologist and professor of instruction at Ohio University. Ann, welcome to the show.
Hi, thanks for having me.
So glad you could be with us. Now, where is Ohio University? Like in real space, where are you instead of just the internet?
So Ohio University is in southeastern Ohio. I'm coming at you from a little bit more central in my basement, but the university is in the southeastern portion of Ohio.
Oh, fantastic. So what exactly is a forensic entomologist? What's the elevator pitch?
So a forensic entomologist is basically somebody, for the most part, and I'll get into this, that collects insects from dead things. And they study insects that come to dead things.
So that's why we had the squeamish warning on the website introduction for the program.
Yes.
I'm with it. Well, I think this is going to be exciting. Everybody, I hope you've got your lunch ready for this one. Dr. Perez, I'll turn the show over to you.
All right, awesome. Thank you all for being here. Thank you all for having me. I'm going to screen share and get my presentation up. I think it's fabulous what the North Carolina Museum is doing here in celebrating insects. And I'm really excited that I'm going to get to talk to you about one of my passions, which is forensic entomology. So I'm going to take you through a little bit of a crash course in forensic entomology, how it works, what insects would be collected as evidence at a crime scene and what you are looking for if you're collecting that evidence, and kind of pepper in my experiences as we go along the way. So just as a general of what forensic entomology is, my elevator pitch is just like one subset of forensic entomology, in that it really includes any intersection between insects and the law, which basically means anything because insects are everywhere and affect people in lots of different ways. But there are three general subcategories. One is stored products, and I don't do these first two at all. So there's stored products forensic entomology, and there's a certain amount of insect parts or whole insects you're allowed to have in food. And so the people who would study those levels, as well as how those insects could have gotten into the food, would be in stored products forensic entomology. You also have urban entomology, which is how these insects can affect live people and their property. So say you bought a house and there was a termite infestation, and you want to know how long it had been there. So if the people who had sold you the house should have been able to take care of it, you could bring in a person who would be considered a forensic entomologist to kind of age how long that termite infestation would be there. And you can imagine that those are really different people and really different in what they study as compared to someone who does this third area, which is arguably the most commonly used area in forensic entomology of medical legal entomology. And that's just going to be studying necrophages insects or death-eating insects that are typically found on remains, but also, obviously, for application purposes, human remains. And these are just some pictures of different necrophages insects. It's a wide world of necrophages insects, and there's a lot of different types. If you were going to go to a body, there could be across its decomposition, over 100 different species that would show up. And if you're looking at just individuals, like the mass of the insects that are there, I have down here over 10,000, but really if you're talking about an adult, average size adult that's been exposed to insect activity, it would be hundreds of thousands of individuals that would be there. But really you can whittle it down, and there's just a couple of important groups that a forensic entomologist in medical legal entomology would focus on. And those groups show up faithfully to a body if it's exposed to insect activity because they use it as a food source, either for themselves or for their babies. So they're going to show up to a corpse to eat tissue, usually soft tissue that's easier for them to break down. But there are other insects that are going to specialize in breaking down those harder to eat parts of your body, like hair and cartilage. Even if the adult's not going to eat once they get there, or on addition to them being able to eat, they're going to oviposit or lay eggs. And those eggs are going to develop into immatures. We're going to see a bunch of pictures of maggots coming up, and they're going to use the body as a nutrient resource as well. It's also the fact that you're going to bring a lot of insects there to eat, a lot of insects of the same species. And so it's a good place to find a mate. So you're going to have mom and dads meet each other at a body as well. Now you do see here pictured the two big groups of insects that are going to faithfully visit because they use it as a food source. And that's going to be flies and beetles. And I will introduce you more to these fly groups later. It's also going to be kind of full of these predators. And these predators show up because there's just like hundreds of thousands of individuals that you could eat. And so it's other insect predators as well as other scavengers like vultures are going to show up. And sometimes we've got insects that just happen to be going by. They're just opportunistic about it. And sometimes there are insects that literally all they do is eat the fly babies that are there. You are probably familiar with the flies that tend to show up at dead things because they're the same flies that show up to your trash, right? These are maggots on one of those big green bins that a lot of people put their trash in. If you throw away food scraps and those are decomposing, same flies are going to come and use it as a food source as well as oviposit. But a forensic entomologist wouldn't be involved in this case. Instead, they're involved in a bunch of other ways to inform legal investigations that I'm going to pop up. We're really only going to cover one, but I thought I'd introduce you to the others. So these insects can be used to determine post-mortem corpse movement. The fact that you've got different insects in Ohio versus North Carolina. If I find Ohio insects on a North Carolina body, you're going to indicate that that body might have been moved. It's also issues of neglect. They do just go to dead tissue, but sometimes that dead tissue is on a live person. And so infestation on a live person could indicate neglect. Typically, if somebody is in a bed, like people who are elderly in a bed and develop some necrotic tissue, they could infest that necrotic tissue. Also, they're going to have characteristics places that they like to lay their babies on a body. It's typically going to be moist places like your eyes and your mouth and your armpits. And if you see them pop up first in areas that are not those places, it might indicate that there was trauma to that area that made it a nice moist place for them to oviposit. But really where a forensic entomologist is most often involved is estimating what we call the postmortem interval. Also, I'm going to use the word PMI a whole lot as an abbreviation. And that's basically estimating how long somebody has been dead using these insects. You can do that in overall two different ways. The first one isn't used as often, but I do love it and I study it. It's insect succession on carrion, and that's just the change in insect species over time. You'll notice my picture up here is of pigs. It's hard to get humans in increased replication in order to study this, so you use surrogates for humans in decomposition, and typically that's going to be a pig. And so early on, you're going to have a specific subset of species that are called initial colonizers, the ones that get there first. Then things are going to show up that want to eat them, or can use parts of the body that are not the soft tissue, so they can arrive later if they're going to be eating head and hair and things like that and be those later colonizers. So what you would do with this information, if I went and studied what insects are there when, I could collect a collection of insects off of a body, right? And so these would each be a different species here, these different shapes. And I wouldn't know the PMI or how long that person had been dead. And I could compare the collection that I made to some data that I collect, right? Of when each species is there. Is it the first day after, the second day after? And really, when you see that same suite of insects pop up in your data set would be the estimate for how long somebody's been dead. And you could get a minimum in the first time that you see that collection of insects and maximum in the last time you see that collection of insects. So those initial colonizers that are going to get there first that we can use are called primary colonizers, like I said, and it's really flies. Flies get there first. And two big groups of flies, Encalophoridae is what they're called, or the blowflies, and Sarcophagidae, which are these guys over here, the flesh flies. You have probably seen some of these bright, shiny green and blue flies, even in your house or in your garage or by your trash. They are the same ones. And then maybe you've seen these, maybe not. They look like house flies. I say they're flies that look like they're wearing a little pinstripe suit, except their butt is a little bit redder if you were looking at the other end. And they both show up very quickly to utilize the body as a nutrient source for their babies. And so a blowfly is going to get there and lay 350 eggs, like lots of eggs. And then a flesh fly might get there and it doesn't actually lay eggs. It does something very cool for an insect in that it just pops out live larvae. It pops up already like living larvae outside of the egg so that they can just start eating right away. Your body is wanted by a whole lot of things to use as a nutrition source and say, I'm going to pop out live larvae that can just start feeding right away. So we really can use all of the nutrition that would be there. Now, how quickly do those actually arrive? And it is in certain condition, minutes. So you place a dead body, expose it outside to insects, and within minutes, they're going to show up. They might not oviposit in those first couple minutes, might take them an hour or so, but they're going to get there very quickly. And you can kind of see them congregating around the head. Like I said, they're going to go for these moister places where they're going to lay their babies or oviposit. So they, as a fly, would show up, lay an egg. That egg is going to hatch, and you're going to get a larvae, what we call a maggot. That maggot's going to grow and shed its exoskeleton multiple times to be a first stage, a second stage, third stage. They call these instars as well. And all of those first stages happen on the body. And so they would be eating tissue here, and you can kind of see their stomach full of stuff in this picture. And then they're going to leave the body. They're going to expel everything that they were eating, and they're going to crawl away from the body because the body is a very dangerous place because of all of these predators. So they're going to find a safer place to go into their cocoon-like stage, which is the puparium. And just like you think for a butterfly or something like that, the cocoon is harder outer shell, except it's called a puparium in flies, or a certain group of flies. So they're going to crawl away, find a safe place, maybe underneath some leaves, and go through pupariation, make a harder outer shell, go through metamorphosis, and then an adult would emerge out of that. All right. So really what we're going to see most often on dead bodies are going to be those maggots right here, right? Maggots. If I showed up to a scene or somebody's collecting at a scene, they're most likely collecting these maggots. And you can see the variation kind of in the size of these maggots. Here's eggs. First stage looks like the eggs just can wiggle around, and then they get really big. So we're talking, almost an inch here. And then this is that hardened stage of the puparium, and then they would emerge as an adult. So you're going to have potentially hundreds of thousands of maggots on a body. And so this maggot, we're going to get into it, looks like this white blob. So if you're actually estimating from just collecting a maggot how long somebody's been dead, you're going to do something similar to what I showed you for succession. We're going to rely on some data that we've generated in the lab. So you're going to have a dead body that I have here. You're going to collect a maggot from that dead body. Then you're going to compare the size and the stage, whether it's at first stage, second stage, or third stage, to experimentally generated data. you're going to actually try and get those that appear oldest, and you're going to age that maggot. And you're going to say, okay, that maggot has been alive for, let's say, 3 days. And so the last time you would have been alive is before that maggot was born, right? So you've been dead for at least 3 days, or as long as that insect has been growing. To kind of show you the data behind it, this isn't exactly how it's done, but this is easier for demonstrative purposes. This would be that developmental data that's generated for a specific species. It shows you 1, 2, 3, those different stages of the larvae and how big they are over time. So as time goes by, they get bigger, except when they crawl away. And it's done at a specific temperature, so this is all temperature dependent. And what you would do is, like I said, you're going to look at something for a specific species. In this case, it's Lucilia sericata. Let's say you collect it and it's 12 millimeters long, right? So if you go, okay, 12 millimeters long, that's about 2.5 days. So this maggot is 2.5 days old, and you would have been dead for about 2.5 days. Like I alluded to, this is species specific, and we're going to have to worry about identifying the species before we ever use this. I also popped up this red. If you did collect something that was 12 millimeters, you'd be like, how did you know it wasn't over here? Well, we can look at the stomach of a maggot. If it's still got food in it, it was still on the body and increasing in size. If it expelled all of that food, it's getting smaller. So you can tell which side we're on by looking at the gut contents of a maggot. So you know that saying that people say, like, you had one job. For a forensic entomologist, the thing you cannot screw up is actually identifying a species correctly, because all of that developmental data that we're using is species specific. And there are species that are like pretty darn closely related and have very different developmental rates. So these look very similar, they're in the same genus, but if you use one species development data for the other, you could be days off in your estimation for how long somebody's been dead. Typically, we don't have those adults that we're using larvae. And adults have all of these like hairs and wings and legs that you can have different features to be able to identify. And larvae don't have any of those. They're like this creamy blob here. There's some on the back because maggots do breathe out of their butts. So they've got these spiracles that they're breathing out of, but there's not that many characters to look at. And so if you come to a body and you see all these maggots, you're like, ooh, I want to try and make sure I get a good representation of everything that's here. But like, I can't tell in this picture if these are different species. So we've got to, when we collect them at a crime scene, collect quite a few, and we want to rear them out. So if you're collecting at a scene, you take what's called companion samples of maggots. You're going to preserve some right away, like stop the clock, stop the biological clock, know exactly what size and stage they were when you collected them. But then you're also going to rear some to adult because the adults are much easier to identify. So in order to be able to rear them, you're going to bring with you to the scene some protein source. I typically travel with liver. Liver is kind of moist and easy for them to break down. So when I take them off of the body, I'm going to give them a nice cozy place to go on this moist liver to develop. And if you look at this picture, this is a great demonstration, but it's actually terrible. I've made every mistake that there is in the book, I think, like most people who practice things. And you don't want to put this many maggots on a piece of liver. There's like 100 and some maggots there. They're quickly going to run through that protein that you just gave them. So you want to bring it down to like maybe 30 maggots that you put on each of your little liver pieces. So this would be way too much, way more maggots there than you would think. Now you're going to contain that protein source. I have it in foil here. I'm making what I call a maggot motel. And you're just separating them from everything around it, because maggots are silly and can't find the food source again if they end up leaving the food source. So they leave and then they can't find it, can't get back, and die. So you want to just say, you have to stay right here on this food source. And then you're going to put that food source, and this is actually cat food. You can always take cat food if you don't have liver or something else. Don't take dog food. It doesn't have enough protein to keep them alive. But they're going to eat here. And then when they're done eating, now they're going to start to crawl away, like I said, to pupariate. And you want to give them this nice cozy place that's dry on the outside, and they can bury themselves to pupariate so that they can become adults and you can identify them easier. So this is a picture, and there is a portion of a dead body right here that is underneath this huge maggot mass. Just to let you know, there's lots of individuals that could be on something dead, and you've got to collect, and you want to collect quite a few because you can't identify them to species right there. You also want to collect those that are oldest. And so you don't know, maybe some slow growers are on here, so you've got to collect. But also those that are crawling away have potentially been there longer because they've gotten enough nutrition and now they're trying to find a safe place to go pupariate. And maggots can do some amazing things. If you look in this picture, they are crawling up a tree. Maybe they're not going to be safer up there, but they go in places where you might not think. I've been to a scene where there were hundreds of thousands on 2 bodies inside a house. And I collected from that. But then you're saying, okay, have any crawled away? Do I have some older maggots here? And so I'm on my hands and knees looking on the sides of the room because a lot of times they'll hit something and try and crawl down, or I'm looking underneath this dog bed that's on the floor. And then as I'm going, it's shag carpet. And so as you're kind of walking, you're parting the shag carpet and they've just gone right down into the shag carpet and stayed there. And so you can find puparium if you just know they're trying to be safe and away from the body once they crawl away. All right, so those puparium that you're looking for, this is about the size that they are. They're most confused with rat poop. So typically crime scenes that I go to are not in houses that are in the best conditions sometimes. And so there might be feces that they could be confused with, but puparia float and poop doesn't. Well, rat poop doesn't. So that's a way that you can distinguish, but typically you can look and tell if you've seen them before. And with those puparia, you're looking to see if they're what we call unenclosed or closed. Unenclosed means that it's still formed. You still got a adult in there that hasn't come out. And then you've got a closed puparia where an adult has pushed its way out and left. And so these can be really important because it says we've completed that metamorphosis and flies have emerged, right, and flown away. So these would be your oldest of the insect remnants that you would collect from those blowflies. All right, so that's generally what you're looking for. On this last one, please indulge me for like 1 or 2 more stories. And I will end with maggot power. I love saying it. There are power of maggots. They do a lot of what I haven't talked about in ecology is just they do a lot of recycling of nutrients back to the earth, but also they can be used in awesome ways like in forensic entomology. And in this picture, you see a couple of things. You've got those other insects that show up just to get some nutrition. But over here, there's actually bone right here. This is the skull of a pig. And then this is the stomach of that pig. You might come here and say, whoa, there must have been some major trauma to that head because look, all of these maggots are here. And there's not really a whole lot of maggots you can see on the rest of the body. But really, this is what you see all the time. You see skeletonization of the head before the rest of the body because that's where they like to lay their babies first. So you see major mass skeletonization of the head first and still a pretty intact body. Or I've gone to autopsies before where you see skeletonization, you don't think there's that many insects there, and they'll open up the chest cavity and then there's all of the maggots that have just kind of moved down from where they've started. Which is a pretty cool observation to be able to see. All right, and so that is my kind of crash course in forensic entomology. And I will stop sharing my screen now.
Thank you so much. Oh my gosh, how fascinating.
I think so.
Well, I mean, obviously that was an amazing presentation. Fantastic job. But wow, I'm kind of in awe of this process. But also the amount of information that you have to know about all of these individual species of flies and their larvae and the maggots in order to draw conclusions that help police investigators, I suppose.
Yeah, I will say I know that people get worried about job security, but if you're going into research anyway, there's still a lot to determine about these forensic insects because they're not always easy to study. And there's so many different situations where you find dead bodies. And so you would want to have generated some data that would be applicable to that situation. And since there's so many different situations, there's so many different types of studies that have to be done.
So I'll remind everybody who's tuned into the program right now You can use the chat box and the comment threads that are on the side and down below the video in order to leave your thoughts and questions. We'll grab those and pose them to our guest speaker today. So, how much of your work as a scientist, as a researcher, is going out and responding to these kinds of calls? Like, are you a phone off the hook kind of thing?
No, not really. So I don't go to that many scenes. I've probably been to maybe 20 scenes, 25 scenes in my lifetime. A lot of what I do is try and train other people so they know what to collect at the scene. So sometimes I do get calls where I'm being asked what to collect, and I'll have a conversation with somebody over the phone. And I do trainings of investigative personnel so they know what to collect and how to um, ship things and send them to me. But it's pretty rare for me to actually go out to a scene.
Okay, okay. But you've done a few of these.
Mhm.
Do you see? Is it all like? Are you always looking for particular critters? Like you talked a lot just now about the blowflies, and I
Yeah,
guess they're a pretty good indicator. They're
yeah.
very helpful.
So yeah, if they're there, you are collecting them. But I really say just get a good representation of what's there. Even if you know something is just there as an opportunistic predator, like you got a spider that's coming over and eating these maggots, right? Even though I know that like that's not going to help me, I'll typically collect it. I want to get a good snapshot of everything that's there. And you never know. You never know what's super important until you actually have a minute to digest things. And so I try and compartmentalize, like when you're there and you're collecting, you're just trying to get a good representation and then worry about what's important once you get back to the lab and start identifying things. But typically, some of the studies that I've done have looked at like what are you likely to find every time you go, because the best insects to then use would be like and try and develop data for those that are reliably there that you're running into really often. And blowflies do tend to be that, right? A couple of different species of blowflies in each area. But I'll tell you, beetles are getting a revival, right? As we've been going through, we realize that beetles are also pretty darn common and reliable in when they show up. So, like I said, people are doing a lot more research with the beetles as well.
What would you say are the limits of forensic entomology right now?
So part of the limits are the the data series that we have generated. Like I said, if you are trying to estimate how long somebody's been dead, you want to use a developmental data set or just a data set that mimics where you collected from. So that's mimics in like geographic location. That's mimics in, is it in a house versus outside or fall or spring? There's all of these different things that can affect the insects that are showing up in their development. So sometimes we just don't have great data sets to compare it to. And also, even for like the developmental data, we have certain species that love to, go through their life cycle in the lab. They're like, cool, I love these conditions. I'm going to go through my life cycle very easily for you. And so we have really great data for those. And then there's some species that hate your lab conditions and they might oviposit and those larvae never go through prepariation and never become adults. Right. And so it's hard to develop data for those. And so we're limited sometimes by the finicky behavior of some of these insects that we haven't figured out yet.
Excellent. Thank you.
Yeah.
All right. There's some good questions coming in here in the chat. So I'm going to throw to some of those now.
Awesome.
Let's see. How do they find the dead bodies so fast?
That's a great question. I would think it is twofold. There are certain chemicals that become volatile chemicals that get in the air as you're decomposing. And they've got some great names like putrescine and cadaverine. They're these chemicals that are named for where they're coming from. And insects can respond to those in certain combinations. So basically, like they've got a great nose for those chemicals that are a byproduct of decomposition that start coming out right away. But then also part of it is like these flies are everywhere as you've experienced through your house, right? So they are typically not going to be that far away from a dead body. So being close and being able to sense those volatile chemicals that come off of a dead body.
Yeah, you had mentioned that earlier about working on scenes inside a home where there were thousands and thousands, which struck me. I was like, wait a minute, but like I think about inside a home, there's not just like enormous colonies of critters.
Yeah.
Unless they're just hidden from me because I'm not dead yet.
So yeah, they're not, I mean, I'll have a couple of flies in my house every once in a while, but
Sure.
once it depends on how well your house is sealed. So if you are in a house that, has some cracks underneath doors or in windows that they can get through, they'll come in. There will be a delay because they're not going to be able to sense those chemicals as quickly. But let's say you're in an abandoned house, right? Or a house that's in pretty bad disrepair, they'll get in pretty darn quickly.
Sure. Makes sense.
Yeah. But there are definite cases that I've seen where people die inside a house and the blowflies actually don't ever get there if you're in a pretty well sealed place. Other insects do. Smaller flies get to you, but maybe not the blowflies.
All right. Let's see. My moderator on YouTube has a good question. Has forensic entomology ever been featured in a novel or movie? And makes reference to the Deathhead Moth in Silence of the Lambs.
Yeah. So a lot of people talk about that Deathhead Moss. So it's basically in the puparium. There's this, or on the back of the back of the adult insect, that there's this like skull looking thing, and in that movie it was like placed there by somebody. It doesn't really have anything to do with the dead body. Somebody would have placed it there, and it just gets its name from the markings on its back rather than like its actual involvement. But yeah, very often it does get featured in some crime scene shows. It does. I haven't read it, but there is a novel that's called like Blowfly. I think it's a Patricia Cornwell, something like that. I could be wrong, but like there are some that really do feature it prominently. I know some of my mentors have been asked questions when they start to write those books, so they'll reach out to a forensic entomologist. But once you read it, they do take some liberties, right? They want to make things a little bit more sexy and fantastic for these shows. So the thing that I run into most often on like a CSI type show is that maggots aren't big and gross enough for people when you look at them in smaller numbers. And they're pretty gross. Like they have to be eating this dead material, and so you're trying to deal with them eating dead material and then say getting them on like a prop body or somebody. Like to actually use those maggots would be difficult. So instead they use like cleaner things that are bigger, and typically mealworms. I see mealworms used a lot, and those are actually beetle larvae that they'll just pop onto somebody and pretend like that's what would be there if you were dead.
Yeah, we had another guest speaking for BugFest, and we had a similar question about like Hollywood bugs that show up. And they talked about a movie. They were supposed to have like some super deadly insect in like this jar, and they were showing off the jar. And entomologists were in an uproar because it was a Madagascar hissing cockroach.
Yeah, which doesn't do anything to you. Everyone can hold them.
Totally harmless.
Oh, that's hilarious.
Yep,
usually,
but they're big.
Yeah, they're big and you can see them and they look kind of gross. So we'll go for those instead. I used to rear insects for selling them for like Carolina Biological. And I'll tell you, it's a very gross thing to deal with them in mass, and it's hard to do. So I can understand why exactly they don't use it. I don't get on their case too much, but the average viewer should know that it isn't real. And just people have different responses. So I would assume that those people who are the actors might not want to deal with it. I'll take live maggots in jars to like BugFest in person so people can feel how much heat they generate. And the responses vary from I'm irresponsible for bringing maggots around people to can my child eat one? So you just have wide ranges of responses to live maggots in front of you.
Okay. That's all right. Let me get back to questions here.
I know, sorry, I got a little off topic.
No, not at all. That's fascinating. Okay, Kate wants to know what happens when the body has been dead longer than the lifespan of the species.
Oh, good question. So, for that species that first shows up, if you've been dead longer and they've crawled away, uh, a forensic entomologist could say you've been dead for, say, at least the lifespan of that species, but it's probably much, much longer. Like, I'll give you this cutoff, but I'm probably not close to the actual estimate. And potentially there's other insects that show up later that are still there. And to say all of the insects are gone, then typically it's not going to be a forensic entomologist that's going to be able to help you as much anymore. And typically you're into like the forensic anthropologist realm. Like then they can help you. So when people talk about postmortem interval estimation, a lot of times like right away the entomologist might not be the best. Like your own body breakdown processes, like your body cooling and your body blood pooling and chemical levels in your eye, those types of things can be used right away in like the first 24 hours. And then the forensic entomologist is typically like, depending on temperature and season, all of these things are dependent on other things, but in general, like after that 48-hour period to, a couple of weeks. And then after that, it's the anthropologist that would be pretty common to try and bring in.
Gabrielle is asking, are the flies that visit corpses the same that visit scat or dung?
That's a great question. So they will visit in order to eat some protein potentially, because they can get a protein meal from that dung. But to actually like oviposit and put their eggs in, no, you actually have I shouldn't say very different, but you've got different species that specialize in dung versus species that specialize in dead bodies among the flies. You've got some beetles that will do both, but the flies are going to specialize on one or the other. So as an example, the sarcophagids, the ones I didn't talk about as much that were the flies wearing pinstripe suits, inside the sarcophagids or flesh flies, a large amount of them are going to be obligate or just go for the dead body, but there are certain species that are the poo loving species. That doesn't mean they can't be used in forensics. That sometimes in issues of neglect, those species are attracted to feces in a diaper. And so if they infest the feces in the diaper, you can give an estimate of how long it's been since the diaper has been changed. So I've never been involved in stuff like that, but there are case studies out there of people, forensic entomologists being involved in cases of neglect that way.
Oh, wow. Interesting. Interesting. All right. Let's see. Glenn says, using multiple species, how narrow a window in time for death can be established?
That's a great question. Again, I'm going to give you like a depends. Typically,
Sure.
early on is where you're going to be able to give the narrowest window. So a maggot goes from egg to first stage pretty quickly. And when you have a high rate of change, you can get a narrower estimate from the observation that you have. So pretty quick rate of change from egg to first instar to second instar to third. But then also, especially because just like your factors, there's a lot of things coming into play. Your estimation gets wider and wider as time goes on and as that insect is developing. And so I can't think about like the number right offhand for, especially when you're looking at multiple, but typically your narrowest range would probably be like a 12 hour, 24-hour range. It's not as concise as you would want. You can't say like it was at 4 p.m. and maybe 5 or 6, but definitely close to 4 p.m. That's not typically what somebody can do. For really early, they might be able to give like a quarter of a day, half of a day, but really not much more narrow than that. We can, however, often say the lower end, like the insects were at least here by this time, right? I can't tell you exactly when they get here, but they were definitely here by this time because they can't develop any faster than that.
So you can come at it from different angles and in the timeline to try to get the best information.
Like I couldn't tell you exactly when it happened, but looking at the range of when this happens for insects, I can give you that lower range and say it's at least this long.
All right. Okay. Have you ever visited a body farm?
I have. Um, I actually used to be able to go with a class, like take students to a body farm so they could kind of see the process. There have been a lot of body farms that have popped up recently. When I say recently, like in the past decade, but I've been to the one that probably people associate with the most, the Anthropological Research Facility at Tennessee, where they do human outdoor exposure, mostly for anthropological research. But a lot of times, if a forensic entomology, say, proposes that they want to do a study, or at least in the past, then they might be able to use the same facility. But I have never done research there. I've just gone and observed.
Glenn was asking if Ohio University has one too.
No, no, they don't. Um, but what I want to develop is just a pig body farm. So not a human body
Mhm.
farm. There's a lot of paperwork. There's a lot of things that you have to go through for accredited places for outdoor exposure of human remains. And I'm actually really interested in developing models that have a whole lot of replication. And so even if I had one, you typically aren't getting like 20 bodies at a time all at once to be able to expose. So if you really want to up the replication, especially for your observations out in nature, then you're going to have to use a surrogate. And so um, I used to spend my summers in Indiana, where we basically had a pig body farm where I would expose about 20 to 25 pigs in a narrow range of time in order to try and figure out what insects were coming and what insects were there when.
Why, Jay's question, why did you want to become a forensic entomologist?
I know, right? Does that say something really creepy about me? I don't know. Is what- so I really just had a love of insects first. I was made to do insect collection in like fifth or sixth grade, and I just went like hog wild. I knew actually I had to do it because I had two older sisters. I had to do it going into that class, so I spent a lot of the summer going into that year collecting insects, and then just had them ready to go when I knew I had to do this. So I loved insects. And my dad was a hunter, and he used to let insects clean, like say the skull or something like that, if he wanted something cleaned. He wasn't going to do it in our house, but he would just kind of stick whatever he wanted in what I call the living fence that we had. There was a bunch of greenery that was our fence. He'd stick it there and he'd just wait for the insects to take care of it. And so I had seen that process. I thought that was kind of cool, but really, I wish there was something better that I could say than I was studying entomology because I loved it. And I was an entomology undergraduate major. And at some point, you're like, what am I going to do with my insect knowledge? Right? There's lots of things that you can, but you've got to go through that process of like, well, what am I going to do with this? And I had a medical legal entomology class, learned about forensic entomology, had reached out to some forensic entomologists, learned that really to be able to do this, you've got to go through research, you've got to understand the process. And so it was suggested you got to get a PhD and study these insects and know them in and out in order to be able to practice. And so I said, okay, I don't know if I'm ever going to practice, but I'm at least love insects and I love research. So why not? And so I went and I found somebody who was doing research in forensic entomology, studied under him, got some opportunities to work with other forensic entomologists, and then got into doing casework. So really not a love for forensics away. Really, it started with a love of insects.
The bugs got to you first.
Yes, the bugs definitely got me first.
So I have a follow up then. How much of the research that you're involved in is related to this forensic work, uh, writing up cases or researching maggots on uh, dead research models versus other types of uh, entomology studies that
I've
uh,
done some other types of entomology studies. So like I've done some other types. I've published, about some other types, but really anything that I do is just forensic application. That is what I want to do. That's what gets me excited. And so all of the research that I've done, um, besides a couple of other areas, is really all about application of aspects of carrion insect biology to forensic situations.
Got you. Okay. All right. More questions here. Oh, Kate's got a good one. How do you personally deal with the grossness of the job?
Um, I honestly think I might have a bad nose. Because I just don't feel like I get as affected by the smell. I can smell it, but it doesn't affect me as much. And it might be because I'm around it pretty often. Um, Sight wise, it does get you sometimes. Like I remember my first autopsy and I got very, very queasy at my first autopsy because of smell, because of sights, the combination of the two. I think you don't know necessarily if you can deal with it until you try it the first time. I'm glad that I got opportunities to like say go to autopsies before I decided this is what I was going to do with my entire life, because I wouldn't have known if I could dealt with it if I hadn't actually done it. So just because you could watch like gory TV, which I cannot watch, I can't watch gory TV, but I can go to a death scene. So like I said, don't know until you get to it, and when you do it, it's if you're like an athlete or you're somebody who does something like where there's a go time, like okay, you now have to perform, or even like as our host, like if there's a go time, you've got to do it, you kind of go into this different mental space where you're pretty darn focused, and you're not thinking about all of the smells and the sights, and you get focused on exactly what you need to collect, and you do realize that it's a pretty serious situation. It's odd because you get excited, right? Because this is what you study and you're actually now seeing it happening. But you balance that with understanding like the gravity of the situation, that this was a person. And so I think kind of like the balance between those two helps as you're going through the process. I kind of got a little off there, but that, yeah, that is
Yeah, no,
that's kind of how I
that's a
deal.
great answer.
You get focused, and you understand why you're there and how important it is, and that kind of helps you put everything else to the wayside. But if anybody is interested in it at all, I would suggest reaching out to like your local coroner or medical examiner. A lot of times you'd be able to observe an autopsy and just see if that's something that you think you can handle. I'll give one other aside. In research and going to the autopsy, it can be quite different. I deal with dead pigs, and those dead pigs do have a distinct smell that is separate from dead humans. And I've run into that a couple times in doing workshops, in that investigators who have been around dead bodies before are now doing these exercises on dead pigs. And I've had one puke before because it was like such a different smell that made him nauseous, that wouldn't have made him nauseous around a dead body. So you can actually have differences there too between doing the research and then, going to a death scene.
John here's got a question about flesh flies.
Mm-hmm.
What happens if the fly cannot find anywhere to place their larvae? Will they just pop out eventually?
Oh, that is a really good question. Yeah, because if you got a live larvae in there, you've got to get it to something where it can eat pretty quickly. I'm assuming that there's got to. I don't know the answer to this question 100%. I don't know if the fly would just let the larvae die, or if it would, I would assume, be pretty non-discriminating about where it puts it. Like it's going to try and find something. Um, and so if it can't find, say, a dead body, it's going to be looking for something else that's a protein source that where it could oviposit. That's a great question, and it's got me curious. I'm not exactly sure. I'll tell you that also it kind of happens in blowflies too. Blowflies can carry around their eggs for so long that they can put one kind of in the canal that's ready, and it sometimes becomes a larvae before it pops it out. So sometimes we have what we call precocious eggs in blowflies, and they'll pop out a live larvae instead of an egg at first because it was holding it for so long. And so if you came to a body and there's a bunch of like smaller maggots and then one that seems to be bigger than the other, it might be that precocious egg that had been sitting in her like urogenital chamber before she put it out. So sorry, I didn't really answer your question, but it's interesting. I'm not exactly sure.
I guess John, not. I think maybe John's been in a lot of the programs, but I've not met John and talked to John up close and personal. John, maybe you should go get an entomology PhD and figure it out.
Yeah.
If you're really interested in it.
It's super cool that it goes through larva position. It's not the only fly that does it. Like the tsetse fly, that causes African sleeping sickness, it like will pop out a live larvae that's basically ready to pupariate. Like it's going to carry it around for a while and then it's going to go out its body and it's just going to go pupariate somewhere. So there's some research that goes into these flies that larva posit, but the flesh flies need help. I will say there aren't enough people who study flesh flies. We have trouble identifying them because there's not that many people who are experts in flesh flies.
When you do talks like this and you've got like a room of people, what's that energy like? Like all of our viewers are online, so I can't see their faces or gauge the reactions, but I could imagine if you're like even at a scientific conference or in front of a room of students at Ohio University, what must that be like when you're like, all right, everybody, today we're going to talk about maggots on dead bodies?
There's variability. It depends on the age group of people that you're talking to and their general interest. So like, let's say in my forensic biology class, right, all of those students where I'm like, okay, we're doing forensic entomology today. They already have an interest and they're studious, they're students, but this is the one time that I have a bunch of crime scene photos that I can use for educational purposes. And they've got this biological knowledge and now they have to like use it as we're looking at these pictures. And man, they are intense. Like this is the most intense I ever see my students is when they're looking at this. But then in like the general public, it's just such a mixture. You've got some people who are like smiling the whole time. They're in awe of it. While other people are just, sitting there very nicely, probably not wanting to be there because maybe it was a little bit different than they thought they were getting into. In general, like at dinner parties or whatever, people are fascinated overall. It's very common that they have a general understanding of what forensic entomology is, but not kind of ever thought or gotten into the nitty gritty. And so like questions are my common thing. Like it does promote question asking.
Yeah, sure.
When you're talking about forensic entomology with any group. So yeah, or like in trainings, like it also promotes a whole lot of questions. I sometimes have to lecture about things that are not forensic entomology, and I'll tell you, I get the most interaction when I get to talk about forensic entomology.
Yeah, I can only imagine that as soon as you start talking about dead bodies, people perk up.
Yeah, people do perk up. There is a fascination with the true crime wave. And then
sure.
yeah.
Well, I mean, it's kind of good to know, hear your stories and then know that you're not dead.
Yeah, it's true. You're like, okay, I'm good. Even though you could have those insects on you if you were alive, but most likely not.
Let's go with not.
Not, yes, exactly.
I prefer not.
It's true. I will also say, if you want to go into this, you've got to have a very either be solo at the time or have a very understanding partner with you. My husband has had to deal with a lot of smells coming into our house because not just if you go to a scene, but also your research all day, you're not the most pleasant smelling person to be around after you're done.
I used to be an animal keeper.
Yeah. Oh yeah.
That I know those smells. Mm-hmm. And so I can only imagine the smells in the line of research that you're in. So
yeah, sometimes I just throw away clothes. We made the mistake of putting some of my really, really stinky clothes after a death scene in our washer and dryer, and our washer and dryer smelled after we were done. I was like, I did not see that coming. It eventually went away, eventually dissipated. But also say I used to have a car that I would carry around dead pigs in, and the smell never went away. On a real hot, humid day, some of those same chemicals would then start to get released from the carpet that were in that car. And although I gave it to my brother-in-law, at some point. That smell, even for him, just never completely went away.
That's the kind of car you give to a brother-in-law, sure.
Yes, exactly.
Yeah.
I sold it to him for $1.
There you go. Okay, one last quick question since it's here in the chat. Novels will talk about applying a salve to the upper lip or nose to help with the smell. Fact or fiction?
That is, so I don't know if I know what a salve is, but like
cream, rub a little something
yeah,
in here.
okay. I've seen Vicks Vaporub used a lot. So many people who are around, even people who do autopsies or are around autopsies pretty often, if they've got what they call a decomp case where you're going to have insects, so it's a little bit older, you've gone through bloat and all of these smells have been accumulating, they do use it. My first autopsy ever, there were 2 people using it and a couple of people who weren't, and I was like, I'm going to be hard and I'm not going to use it, I don't need it. And I wished I would have. And since then, I've used it one time, but typically I don't. But like I said, I do see it used by other people. So that I would put in the fact column.
Awesome.
Yeah.
And excellent presentation. This has been fabulous.
Awesome. Thank you.
And everybody in the chat is dropping their appreciation as well. Just so you know, they've all loved this too. Just a great show all around. Great stuff.
I'm so glad that you've got people interested in insects. We need more of them.
You know, for BugFest, I think I saw we had thousands of registrations for our programs throughout this week. So it's been a fantastic week. We've had all kinds of great conversations with people all over. So,
awesome.
It's been a great BugFest for us. Thanks for being with us and sharing your knowledge and expertise.
Well, thank you for being awesome too. Thanks to the North Carolina Museum of Natural History, and thanks to everybody who's interested in insects. You guys are awesome. Stay curious.
That's it. All right, everybody, don't forget, we still have programs coming up for the rest of today and all day tomorrow. So hit up bugfest.org. Many thanks to our BugFest sponsors, Terminex and BASF. You can go to naturalsciences.org, which is the museum's main website, to get information about our upcoming Open on September 22nd. That's next Tuesday, 10 a.m. We are excited to welcome everyone back to the museum. You'll need a free timed reservation, but you can get all that information as well as read up on our safety protocols that are in place and our change in the hours of operation at naturalsciences.org. It's right there on the homepage. So we'll look forward to seeing you back in the museum very, very soon. Until then, follow the museum on social media. We're on Facebook, Twitter, and Instagram at Natural Sciences. And you can subscribe to the museum here on YouTube in order to see when the next live programs are coming up. Click the bell to get notified as well. That way, when we're bringing you great science stuff out of the museum, you'll get that little ping and you can come and join us for the next exciting program. Thanks, everybody. Oh, and don't forget, go buy a BugFest t-shirt. They're really cool looking, and you can get whichever color you like. We have a whole rainbow of colors. So, okay, I think I said it all. Thanks, everybody. Take care. Stay safe. We'll see you again soon.